fish and other vertebrates have similar immune systems to us in terms of they have adaptive immunity as well as innate immunity. So these things are, it's quite fascinating. It goes back a really, really long way in terms of the tree of life. And there's a lot of crossover and there's a lot of conservation of immune mechanisms between fish and sharks and us. If we can get these eDNA technologies to a point where we can use them to quickly and accurately monitor biodiversity, I mean, that is really huge. We need to be making decisions around marine conservation quickly and in a way that can, protect our oceans, especially since they're facing so much threats from climate change and things like that. So I think, at least from the eDNA side, that's a huge thing. And this also means that, even just beyond our lab and our local area, we can, make sure that we're supporting local communities, Indigenous communities, by providing these tools that can, enable better protection for culturally important sites and species in the.
Welcome to a new episode of WeScience. At WeScience, our goal is to promote local science on a global scale. Today is going to be an exciting chat with my colleague, Dr. Emma Jong. She is a leading bioinformatician at the Minderoo OceanOmics Centre at the University of Western Australia here in Perth. And I'm very excited to talk to her because she has skills right from wet labs to dry lab skills. So welcome, Emma, to this podcast. And I'm really, really excited to talk to you about your experience, your research, and what is your perspective for the future. Thank you.
Thank you for the invitation, Carlos. I'm really excited to talk to you today.
Great. So I just wanted to start from the beginning. What motivated you to become a scientist?
I was thinking about this in preparation for this, and I feel like it has to be linked to the fact that both of my siblings had a childhood cancer. So my sister had an acute lymphoblastic leukemia, ALL, when she was very young. And then my brother had a Hodgkin's lymphoma when we were teenagers. And they're both fine now, both healthy now. But I think, going through that experience always made me really curious about human biology and disease. And I think particularly immunology, when I was old enough to realize that my sister's disease was like a blood disorder and things like that. So I think that's where that came from. Yeah.
Wow, that's an very inspiring story because, I have been talking to some colleagues and friends here in this podcast, and then some have shared that they had that previous experience of having a close relative being diagnosed with cancer, which is a terrible disease. And we share the same interest in terms of cancer research.
I think it can be really motivating. Yeah.
And also because, for example, acute lymphoblastic leukemia is the most common cancer in children.
I was always really curious as to what actually happened. Like, why did she have too many white cells and what was going on there? So I think that's probably why, in particular, I really like immunology as well.
So how that motivation and inspiration continue you in regards to your academic journey?
I think that, for a while there, I considered doing pharmacy, which I'm very glad I didn't do now. I think when I was deciding what degree to do after high school, it just ended up being a pretty stock standard Bachelor of Science. It was broad enough and it was just what I was interested in. So I just went with that without really thinking about the career that I would have on the other side of that, to be honest. So my path in that way has been pretty straightforward down like the academic pathway, I would say. So I did my Bachelor of Science, and then probably when I started to learn more about immunology, microbiology, infection, I knew that that's what I really was interested in. So I focused on that as my major, and then did a 1-year honours research program in immunology and pathology. Right after my bachelor's degree. And then from there flowed into my PhD. So one of my honours examiners became my PhD supervisor. So it's been a pretty straightforward journey in that way.
So this PhD is from Murdoch, right?
Yeah, from Murdoch University.
So could you please share about what was your PhD about?
Yeah, it's going back a few years now. I had a look at my thesis to prepare for this. I was like, wow, that's a long time ago. Yeah, so my PhD thesis was in the field of preterm infant immunology and looking at how one particular immune cell monocytes respond to infections. Babies that are born really preterm are at a higher risk of developing sepsis or blood infections. And even though it's sort of generally recognized that their immune system is immature, but there's still a lot that we don't know about exactly in what ways it's immature and how this links to why they get sepsis. So my PhD was very highly focused on this one immune cell from the innate immune system. And we thought that for preterm infants, that their monocytes would not respond as well as a term infant monocyte to bacterial pathogens that cause sepsis. But in the end, we found that that's actually not the case. So I did some sequencing, I did some flow cytometry, lots of different things. And essentially what we saw was that preterm infant monocytes actually have everything they need already to respond to bacterial infections in a similar way to a term infant. So the only thing that is slightly different was that if a preterm infant had been exposed to infection before they were born, their monocytes behaved a little bit differently. Other than that, they were fine.
It's very interesting in regards to preterm and on term, right? That there is no much differences in that regard.
Yeah, I mean, it was not the result that we were expecting at all. So, and it's funny because I was I'm not disappointed, but when you get negative data, it's somehow not as exciting, but it's still a really important result because it shows that that's not the place to be looking. So,
And you bring a very valid point in regards to the negative data, because as scientists, we wanted to always have positive data or data that is significant. That is great because we can advance, and because basically we are testing different questions. And as you said, whenever we test a question and the question is negative, it still brings information and it still is important. The system sort of lead us to, and also ourselves, to be able to focus more on the positive
exactly.
data.
It's a bit of a problem, I think. I mean, I feel like negative data is equally as important as a positive or significant result. So,
if, for example, we get a negative result, then if we don't publish that data, then other scientists will dedicate some resources and time to find the same answer. So it's important to publish that data, to bring value to that data, because otherwise we can maximize the resources.
The resources. Yeah, I 100% agree. 100% agree. And it shouldn't be difficult to publish negative findings, I think. It should be as equally easy to publish as, a significant positive result.
So, so you were mentioning about your PhD about sepsis. Could you please describe in lay terms why sepsis?
Yeah, so we focused on sepsis because preterm infants are highly susceptible to sepsis. So the earlier, the more preterm, the higher the risk of an infection. And some of these infections are life-threatening. So the mortality rate for late onset sepsis, I can't remember the statistic now, but it's much higher the earlier preterm you are. So it's a big problem. And the interesting thing about preterm infant late onset sepsis is that some of the pathogens that cause disease are things like Staphylococcus epidermidis, which just live on our skin as adults. So they're typically considered not pathogenic, but in this vulnerable population, they do cause severe disease. So it's quite an interesting, it's quite interesting, and it's a huge problem.
Right. And then you postdoc to the kids and work on bioinformatics and data science and computational biology. Could you please describe, before we jump into that, could you please describe on lay terms what they are?
So I suppose probably to explain my transition a little bit, during my PhD, one of the main things that I did was use RNA sequencing to assess the monocyte responses. So at that point, I was really transitioning from wet lab into learning about sequencing and how to analyze that data. And so that's kind of what I ended up focusing on a lot in my postdoc. But I think in very simple terms, bioinformatics is just using computers and software to store and organize and analyze biological data, like DNA sequencing or RNA sequences. And I think the other one you asked me to look at was, how do I define computational biology, which honestly I think has a lot of overlap with bioinformatics and probably just includes aspects of both bioinformatics and data science to, inform about biological data. So I hope that's a simple explanation.
Oh, that's fantastic. Yeah. So why is it important to use those transcriptomics tools to be and bioinformatic approaches to dissect the role of immune cells?
So from my perspective, I think transcriptomics is a really fun layer of biology to study because it's kind of this dynamic layer that reflects both the underlying genetic DNA sequence, but it also captures changes to environment and how the cells are responding. So for me, it's kind of like this really like information rich layer of biology that we can use to understand what's going on inside a cell. So it's sort of like this middle ground I would describe transcriptomics as. So I think that asthma is a really good example of how we can use transcriptomics to better understand disease. So asthma is actually a really complicated disease. It's not even a single disease. It's like an umbrella term that encompasses a lot of different subtypes and different sort of asthma mechanisms. One thing that I found really interesting through my postdoc is even though we know this, we know that asthma can be sort of allergic or non-allergic. It can be triggered by viruses or by genetic or other environmental things. The treatments for asthma are basically the same across everyone who has asthma. So that's quite interesting. And I think more recent studies that have looked at transcriptomics approaches to measure gene expression in cells, either from the lung or from the blood, have shown through sort of unsupervised clustering that there are many sort of different hidden subtypes of asthma that, that patients can be grouped together by their molecular mechanism, not just their presentation and their symptoms. So I think this is really important because when you start drilling down to that level and looking at the molecular pathways that are active, then you can start to find treatments that are really specific for that group of people. And I think that's where we get the power of transcriptomics to help look at disease mechanisms and therapies.
Great. And also you work on cancer as well. You have a lot of publications on cancer.
Yeah, so I did a lot of collaborative work in the field of cancer, some to do with immunotherapy. And I also worked with a colleague and a friend on looking at how natural killer cells can be used to battle leukemia. And yeah, I think that's honestly a bit of the nature of being in bioinformatics is that you end up collaborating on a lot of different projects. So that probably explains why my CV is a little bit diverse in that way.
No, because it's great because you have the skills to be able to transfer across the different diseases, right? So you develop a very strong skill set of bioinformatics
Yeah,
tools.
I think at the time, sometimes I was worried that I was becoming not a specialist in anything because I was like a little bit of asthma and then a little bit of cancer stuff and a little bit of infection. And I think coming off the back of a PhD where you feel very expert in your one thing, sometimes you can feel like, oh, now I'm not actually an expert in anything. But in hindsight, I probably was becoming an expert in the bioinformatics space, but I just didn't really think of it like that.
Because bioinformatics allow us eh, to be able to sort of interrogate, questions, as you were saying, in terms of the immune compartment, to be able to understand the different cells as well as the different pathways or cellular networks that are sort of activated in the particular pathology.
Yeah, absolutely. Yeah.
And also, it's like, for example, you were talking about bioinformatics and computational biology. So you were talking about like you need to have that level of data to be able to sort of dissect the complexity of the cells, because cells are not just RNA or protein or DNA. It's very complex, and that's why there are different fields. For example, in the case of system biology, where you can apply not just understanding one layer of complexity, but it's also understanding the layer in relation to something, right?
Yeah,
So you are
absolutely. Sorry, you go.
so what are the main challenges in terms of systems biology approach?
So I would say that, I mean, obviously you're dealing with just a lot more data, number one. But I think that sort of we have this image of how all these layers of biology are coordinated and they talk to each other, and we can use all of them to get a really clear picture of what's going on. But I think one of the challenges is that these different layers don't always talk to each other in the way that we think or in a simple way. And so, for example, there are lots of examples of where gene and protein levels are not in sync. They don't correlate. And this could be for a lot of different reasons, like timing prepackaged proteins already in the cell and things like that. So I think that's a challenge is sometimes we might expect that certain layers of biology will sync up and show us a particular thing, but often they might not. And it's how to deal with that. And I think another challenge is just when you have all of these data sets, how do you integrate them and make the most of all of them? So it's been a while since I worked in this space, but I believe there are some good bioinformatics tools now for integrating multi-omic data sets. And I'm sure that will just keep continuing as well. I would probably, one of the biggest things is just the amount of data.
A person who might be listening or watching this podcast might say, wow, how the data comes from. And particularly we are like a sequencing, basically with profile. We use genomic tools or omic tools, as Emma was saying, multi-omic tools, which omics means that we are profiling everything. So all the proteins, all the genes, etc. So depending on what we are profiling, if it's DNA, that will be genomics. If it is RNA, that will be transcriptomics. If there is, it will be proteomics. So then all those different layers will be generating a lot of data. One of the challenges is because it's a large data, and then how we're going to digest that data. That's another question. And then you have to use bioinformatic tools to be able to make sense of that data, to be able to reduce the amount of data. I'm talking like a expression of 20,000 genes in one cell, and then you had to account the different cells, etc. So you had to add all that in. So that's large amount of data that you had to analyze and be able to reduce to something that we can understand.
Absolutely. But I actually think another really important point to make is that this is where having a very focused analysis plan or a hypothesis is so important, because I feel like it's very easy to become overwhelmed with the data that you have, and, wanting to look at it in a million different ways. I do think discovery science is very powerful, but I think if in certain situations, if you don't keep it focused or come back to your hypothesis, things can become unfocused very quickly and it can be very overwhelming. So I know it sounds so simple just to come back to your question and your hypothesis, but I mean, I've even been there myself, and that was some advice that I got during my PhD and it's always stayed with me. Yeah, I think it is so important.
I guess one of the complaints that dry lab scientists have in relation to wet lab scientists is that wet lab scientists, we think about the experimental design, but the less that we think is about the analysis, how it's going to be analyzed. And that's, as you were saying, you rightly pointed, is like you need to have an hypothesis. This is not just to shoot arrows everywhere. It has to have an
Yeah, absolutely. Yeah. And it comes back to that, this is not anything new, but just having both sides, like wet and lab, together in the same room to talk about experimental design and to have everyone present from the beginning, I think is so important. But, this is something that I think people that work in bioinformatics have been saying for a very long time. I don't think it's getting better, to be honest. I think bioinformatics is becoming so much more integrated now into wet lab stuff as well.
And that's fantastic. But still, I guess that challenge in terms of the analysis, right? In terms of be able to reduce the information to something that could be actionable. That it could be used as a treatment or as a biomarker, etc.
Yes, definitely. That is a huge skill. And I think that even though it's very boring, things like proper data QC and cleaning data and making sure your data is very accurate plays a huge role. And I think it's very easy to get excited and want to skip over a lot of that stuff and just look at your data downstream. But spending a lot of time in those early stages, I think, are also really important. It's just not the funnest thing to do, but it's so important. Yeah.
We faced the situation when we were working on single cell RNA sequencing, where we had the samples ready, and then once we spent a lot of money, we realized that the samples were not right, were not okay. The quality was very low. And then you have to repeat it, and that's more money. So it's important that, as you said, is be able to have QCs at every time point of the process, not just
absolutely.
QCs in terms of the sequencing, but QCs in terms of the wet lab part, the workflows. The quality is high, because if you as a wet lab scientist say, oh no, the RNA is sort of okay. Sort of okay, which show, as they say, rubbish in, rubbish out in terms of analysis of data. Yeah.
I think, you raise a really good point. That's like QC along the whole workflow, not even just the bioinformatics end, but through the whole workflow. I think it's so important. Can save a lot of time and money by doing it right. Of
course, no. We spent a lot of money on just that. And it was not because of the cells were because they were frozen. The cells were
Hmm.
okay. When we sequenced, they were okay. So that was the problem. It's no that we were not careful about the cells. It was that
Yeah. Yeah.
we couldn't use the frozen cells to be able to as an input for the RNA sequencing part.
Hmm.
Yeah, that's part of the learning. For some samples, it works, but for others, it doesn't.
Yeah. Yeah. That was very frustrating.
Then you were working on a very strong bioinformatics scientist with lab experience. And then you were very, successful in other kids. And then you decided to move to marine biology, which is very exciting.
It is.
And like the oceanomics part. And yeah, so how was your transition? What made you move from this comfortable space, which is applying your bioinformatics skills to any
Mm.
particular place, and then be able to sort of jump and say, hey, I'm gonna use multi-omics tools or genomics tools to be able to sort of bioinformatics approaches to be able to sort of understand and profile marine
Yeah, it sounds a bit mad sometimes. Well, I feel a bit mad sometimes that I've made such a huge jump. But yeah, I feel like the transition from human medical research to now working at the Minderoo OceanOmics Centre has been great. I think probably in terms of my motivation, I was really in my comfort zone. After I think it was like 6 years or so with the Kids Research Institute. So yeah, I was doing a lot of the same stuff like over and over again, which is fine. Like there's nothing wrong with that. But for me, I was sort of at a point where I was like, I really would like to expand my skills, learn something new. I just felt like it was probably time to get out of my comfort zone. And I always knew that if it wasn't human medical research, like environmental science is something that I was also passionate about and thought that, this would give me a lot of job satisfaction. So it almost was just a bit opportunistic as well. Like the opportunity came up, so I went for it. But it was very difficult, I have to say. Like, you're almost starting again in a lot of ways. I had to learn like very basic concepts about genomics and genome assembly, and even start learning, coding languages that I'd never really had to use before. So I really felt like I was starting again. And that's hard. Like that's really hard because I've gone from feeling like I'm an expert and I have all this expertise and people recognize that, and I've got all these collaborations, and then just kind of moving into this completely new space where I don't have any of that. Yeah, it was very challenging, but it was good because I've learned a lot in the last couple of years. Yeah.
Yeah, you are very brave, Emma,
Yeah. Thank
in
you.
jumping. Not only
Well, crazy.
other people stay. No, because other people will just stay and make your name and become like a global leader in the particular field, and you were in
Yeah.
the path to do so, working out across different diseases, etc. So it's great, but
Yeah.
jumping into a new field, which is marine biology, is something that
Hmm.
it might come with a lot of challenges. So you're leading a bioinformatics teams at the Minderoo OceanOmics Centre. So could you please share your current research in lay terms?
Yes. Yeah. So my position as the lead of the team is quite new. So that's very exciting.
Great.
And I guess describe the work that we're doing at the centre more broadly. So we're really focused on two main things. So the first one is developing technologies that use environmental DNA or eDNA, which is just all the tiny fragments of DNA left behind in seawater from all the animals that live there. So developing technologies around how we can use that information to monitor biodiversity. This is a super exciting field, I think, because it can really offer like a hugely scalable and high resolution picture of the biodiversity in our oceans, especially when you think about how this is traditionally done with like visual surveys and things like that. Like we really need these like better scalable high resolution techniques. So that's a huge focus of what we're doing at the centre. I will say, so the other main focus that we're doing is developing a library of high quality reference genomes for marine vertebrate species. And that's because currently one of the limitations to eDNA technology is that we have sort of a lack of reference material to map it back to. So the other huge focus of our team is building up this library of reference genomes for marine vertebrate species. And we want to make these all publicly available so they will help with the eDNA side, but they'll also be just resources for other people to use for anything downstream that they want to use. And so then alongside that, I've also been sort of dabbling in a comparative genomic study looking at the immune system genes in some of these vertebrates. And that's really just because I would love to bridge my two fields now. And we have the data. So yeah, why not have a look?
Great. So I'm very interested on the environmental DNA part. I guess, correct me if I'm wrong. So imagine this like a soup. So the ocean is like a soup, and then you're able to identify the different species that are there based on the DNA that's shared by them
Yeah.
every day. And then be able to detect what will be the species that are circulating in a particular area. Because it has a dilution factor, it will be very diluted to be able to concentrate the DNA and also be able to profile it and be able to sort of have the catalog of the atlas of the different species that are there. Wow, that's mind-blowing.
Yeah, and like you're right there on like that's kind of one of the challenges as well, is that, you know, it is this soup and it's not just vertebrates that are shedding their DNA. There's a lot of bacterial DNA that we have to deal with as well. So it's very technically challenging, and there are a lot of things that we're working on to try and improve the sensitivity of these assays. So for example, we can't often get all the way down to a species level identification from the eDNA data. We can get it to genus level, for example, with, you know, reasonable amount of confidence, but it would be fantastic to make that even more sensitive so we can tell exactly which species are or have been in a certain location.
So we have to be very careful when we go to the ocean, I guess.
You will leave behind your DNA. Yeah, we all contribute. Yeah.
So wow. And then how you are able to detect those fragments?
So there's multiple stages. The first stage is like an amplification. So we extract the DNA. If I get this right, we filter liters of seawater through a filter to try and capture and concentrate all of the material onto that filter. And then the DNA is extracted from that filter and amplified and cleaned up. And then we use a like metabarcoding approach for the sequencing. So we're targeting specific regions that are conserved across marine vertebrate species with enough variation on either side of that conserved region to then make a species level detection if possible, or a genus level detection. So it is targeted. We're not just sequencing every single thing that's in there. We are trying to target specific taxa with these assays.
So is there particular like an endangered species that you have the DNA that you wanted to sort of understand?
In terms of the eDNA or for the reference genome side as well?
The eDNA.
That's definitely something that is an application of this technology, because if we think about this as it's a non-invasive sampling technique that we can use to get a picture of what species have been where. The thing that would limit it is making sure we have matching reference material for those endangered species. And sometimes that's difficult because, you know, they are endangered and they are difficult to find, difficult to sample. But yeah, absolutely, using eDNA can be, a great way to non-invasively track and measure how like all species are going, in particular endangered species.
Yeah, in one of the previous episodes, I was talking to Arutha, who is a leader on spatial transcriptomics as well as a liquid biopsy. And one of the issues that he was saying is in terms of detecting cancer markers in the blood, one of the issues is dilution. And then for this particular one, in terms of the ocean, be able to filter, to extract, and to profile and detect what you really are looking at. So that is, you know, it has a lot of work behind that.
Yeah, definitely. And I think there's a lot that can still be done in terms of, developing the assays to try and make them more sensitive as well. So, it's a very exciting thing to be involved in.
And you were talking about that you are very interested in bringing your two passions, immunology, genomics, and marine biology. How is the immune system in terms of fish related to humans?
It's so interesting. I mean, this is something that I've spent a bit of time reading and learning about as well. Fish and other vertebrates have similar immune systems to us in terms of they have adaptive immunity as well as innate immunity. So these things are, it's quite fascinating. It goes back a really, really long way in terms of the tree of life. And there's a lot of crossover and there's a lot of conservation of immune mechanisms between fish and sharks and us, for example. So it's kind of nice because I feel like I can sort of, bring these two worlds together a little bit.
But that's great. That's fascinating.
Yeah.
So you were also talking about these reference genomes, and some people will say, what is the reference genome? And when the Human Genome Project, they finished with the sequencing of the first human genome, we were able then
Mm.
to be able to sort of contrast and compare the sequencing that we are detecting compared to the reference. And that's what, I guess, what you guys doing in terms of you had to get DNA from the species, marine species, and be able
Mhm.
to profile them and then related to the species, and then be able to sort of develop its own reference genome, isn't it?
Yes, absolutely. So I think, and that's one thing that I have really realized leaving the world of human medical research and going to sort of an environmental science is that, I really took for granted all of the data and resources that we have available for humans and for other model organisms. It's just so lacking for so many other species. And so, we're a part of this effort to improve that and provide high quality references for as many marine vertebrate species as we can. But, there are actually so many other groups in Australia and worldwide who are doing similar things. So we're part of this really large international initiative called the Earth Biogenome Project. And their mission is, I think it's something like to sequence and catalogue all life on Earth. We also collaborate a lot with people at the Darwin Tree of Life program in the UK. So they're really focused on sequencing and cataloging genomes for species across the UK. And then we also work with the Vertebrate Genome Project based in New York, and they're focusing really on vertebrates. So there's a lot of work happening in this space. And yeah, like you say, it's really about providing that reference material that can then enable so much downstream research in these non-model organisms.
And it's important to know, what we have. We need to
Yeah.
have like information databases of what we have so we can preserve and conserve those species.
Yeah, absolutely.
Yeah. So what are the main challenges in terms of, you were talking about the reference genomes? I guess you had to use genome assembly. So what are the main challenges of using genomic tools to catalog marine species?
I would say that, so at least in terms of the bioinformatics workflows, it's really easy now to assemble a genome, at least for fish. Some of the larger, more complicated genomes, for example, some of the sharks that we've sequenced, it's a little bit trickier, but overall, the bioinformatics tools are really, really good now. So I think some of the challenges, though, the caveat with that is that it relies on having certain types of data, particularly long-read DNA sequencing. So for us, that means that we use the PacBio hi-fi sequencing as sort of the backbone of our assemblies. And I feel like that's really critical. So that's a challenge because that type of sequencing is relatively expensive, and you do still need quite a lot of high quality DNA for that to work effectively. So probably that's the challenge, is around getting enough high quality DNA from the samples. And the expense, I think, is still a barrier for a lot of people.
Yeah, you were talking about the long read, and there, for people who don't know much about the genomics part, is that you have that mainly two types of sequencing. One is short read, and the other one is long
Hmm.
read. Basically, short read is be able to, if we have this length, we just need to sequence. We sequence these pieces, and then we be able to assemble them, and then be able to build the the reference genome from that.
Yeah.
The advantage of the long read is be able to sort of sequence, instead of sequencing small pieces, we just sequence large pieces. And then it's easier to be able to assemble against the reference genome.
Yeah, it's like you're putting together the puzzle with larger pieces already put together. That's like an analogy, I guess you can use. But there was sort of a concept that longer read sequencing was a lot less accurate than short read. And I think that probably was true in the past, but I think that's changing. So longer read sequencing is now becoming very accurate, which is fantastic. And hopefully will become, or slowly become, cheaper as well.
So what we gain from this research?
What do we gain from this research? I feel like if we can get these eDNA technologies to a point where we can use them to quickly and accurately monitor biodiversity, that is really huge. We need to be making decisions around marine conservation quickly, and in a way that can, protect our oceans, especially since they're facing so much threats from climate change and things like that. So I think, at least from the eDNA side, yeah, that's a huge thing. And this also means that, even just beyond our lab and our local area, we can, make sure that we're supporting local communities, Indigenous communities, by providing these tools that can, enable better protection for culturally important sites and species in the. And I think on the reference genome side, is having these resources is beneficial for so many things downstream. So a lot of these species are important for fisheries, supporting local economies, like people who need to use these resources for population genetics, for example. Yeah, better understanding fish stocks and sustainability from a recreational and commercial point of view, I think is really important. Yeah, and I think, like I mentioned, we want these reference resources to be publicly available for anyone to use. Yeah, there's just so much that can be done with these resources. Evolutionary biology is another example, understanding how species have diversified. Yeah. Yeah, I feel like it can be used in a lot of ways.
And particularly, we were talking before, is that we cannot conserve what we don't know. And
Yeah.
particularly, we looking at the ocean and we just take it for granted. But without the marine life, humans cannot exist because
Hmm.
so there is all these interactions between the different ecosystems, etc. So
Hmm.
whenever even we are putting like a plastic bag or whatever in the ocean, that might affect the whole ecosystem, and that might affect different ones, and then we can have a big impact. So if we started to fish endangered species, we are able to destroy all those different ones, then, our life, our time on the earth, it will be shortened because of that. And it's important, all the great work that you guys do, to be able to sort of understand, catalog, basically understand
Yeah.
what we have so that we can have better strategies, right? To
Yeah.
prevent.
Yes,
Yeah.
absolutely. Yeah.
So then how the Western Australia science, how you guys
Mhm.
plan to influence global marine biology research?
I feel like so WA, we're unique in that we have, very rich biodiversity off our coast, right? So by building these resources and applying them locally, we can obviously firstly catalog the rich biodiversity that we have. That's obviously hugely important on its own, but then we're also contributing to these global data sets and databases in terms of both the environmental DNA data and also the reference genomics data. And some of that is even to do with method development. So any DNA methods or advances that we can make can be used by anyone anywhere in the world as well. And the same for the reference genome side of things. And so I think that's why, when I mentioned before that we're working with these other global consortia, it's so nice because these other consortia have spent a lot of time and effort in developing these technologies, and it's such a collaborative working environment. So we have regular meetings and seminars that we can attend together. Everyone is very happy to share and help each other. So I think that we have hugely benefited from what others have done in the world, but similarly, anything that we develop and any sort of like advances that we can make will also help people doing similar things in other parts of the world as well.
Yeah. And also, for example, what you said is like in Australia, we benefit from having the technologies, the genomics, the sequencers, and all this. They are expensive. They are not cheap.
Hmm.
And we develop these low cost or more implementable tools will benefit countries with lower resources, that they have an interest in the understanding the diversity that they
Hmm.
have in oceans. Particularly, for example, I'm from Colombia, so there is
Yeah.
a lot diversity in the oceans and the Pacific
Yeah.
as well as the Atlantic Ocean. So it's important to sort of develop those tools to be able to sort of be deployed in these low resource countries.
Yes, absolutely. Yeah. And that's why I think it's so important to have resources and bioinformatics tools and all of that stuff just be freely available, publicly available for others to use and implement, which I have actually always really thought was nice about the bioinformatics community in general. It's very collaborative, I think, and people are often really willing to help and share, it's just a nice way to work. Yeah.
So your skills, and where, as we were talking in this conversation, so you went from well lab and dry lab skills in
Hmm.
human, then you jump into marine biology. So and you share about the challenges that you face in terms of moving.
Yeah.
How do you think? And I'm pretty sure that your skills were able to advance because some information and tools that are already for human research can be deployed
Hmm.
into biology research. How do you see the marine biology research that you're doing to sort of benefit human research?
Well, I love this question. I think this is a really good question. And I think you're right in that there is like this, well, there can be this like two-way exchange between human research and other research, whether it's marine or some other system. And the example that you gave in terms of genome assembly, I think is the perfect one. So that was obviously established and like a lot of progress was made because of the human reference genome project, but we can now use those tools and resources in the marine space. So that's like a huge example. I think going back the other way, probably from like a wet lab point of view, some of the samples that we work with are really tricky, like either degraded, low quality, low concentration, just whatever we can get from the environment. So I think that in that space, developing, and optimizing methods that can work with these really tricky materials can definitely benefit the medical space and the human space because we can't always get perfect samples in human medical research either. And I think on top of that, if we're like even zoom in more on like looking at the immunology and cross comparing that, I think there's a lot that we can learn. So even just in terms of like the evolution of the immune system, for example, if we can compare like us with fish and sharks and other ancient vertebrates, we can tell which parts are highly conserved, which means that they're probably very critical for all these pathogens. And we can also learn which parts are more specialized to us, for example, or how this relates to our disease vulnerability. I've actually been reading recently about sharks, and they have this, they're part of I think they're one of the oldest vertebrates that have adaptive immunity, which is super interesting. And they have an antibody that we don't have, and it's very different in its structure. I think it's called Ig new antigen receptor. And basically people are now looking at either using this molecule or synthetically making this molecule for treating human diseases. I think there's like direct things that can compare across, but also, just more generic things in terms of method development as well.
Great, that's fascinating, right? So as scientists, we have lots of failures and some little successes. So could you please share some of those most significant ones?
Yeah, I was thinking about what I would say for this question. I feel like, honestly, I don't know if I've had too many huge, like, eureka moments. I feel like probably I have a lot of little ones, like, especially to do with code, debugging code, things like that. I feel like they're like daily challenges and like little daily wins. I have had some grand success and all that kind of stuff as well. But yeah, I just feel like I don't know if I've had any gigantic moments like that. And then thinking about the failures, I mean, I feel like there are a lot of examples of failures. Through my PhD, I spent a lot of time trying to optimize a protocol that just never really worked. So change directions. I've had many grants rejected, things like that. So I think, all of those things are failures, but I think that what I take away from that is that there's always something positive that comes out of that. And I think, especially for like in the context of the lab, I honestly feel like you become a better scientist when you have things that fail in the lab and you have to think about what's going wrong and how to troubleshoot it and how to fix it. I think that's such a hugely valuable learning experience. So it's hard to really see that as a failure. And then even in terms of grants that have failed, that obviously sucks. But I think that even though you've spent a lot of time doing that, you've probably come out of it with maybe stronger collaborations with the people that you're working with. All that writing you can repurpose for something else. So yeah, I think I just like to think positively about it. I think there's always something you can take away, even if it feels like a failure at the time.
We have to be very positive and optimistic and also very resilient, right?
Absolutely. Yeah. Yeah. You can't let it get you down.
So what factors, Emma, do you think contribute to your success in science? As scientists, we have different challenges and we have also, a supporting environment. So please could you highlight some of those?
Yeah, I think that contribute to my success. So definitely having good leaders and mentors, I think is really important. Like I know this is something that people have said to me, or even I've been in, sessions where people are giving advice to younger researchers and it comes up a lot. But I think that for me personally, a lot of my mentors, I didn't find through a formal mentorship program. They're just people that I have either worked with or come across in my career that you kind of naturally develop these relationships with. And they're people that you can turn to for advice and who will advocate for you or help you advocate for yourself. So I think that having those people is really important. But, you probably have them and you don't even think of them as a proper mentor. But, I think that is super important. And then also collaboration, like we don't ever do research just by ourselves. I think the best science that we can do is when we collaborate with people across disciplines and across teams. So I think that, yeah, just collaborating with people as well is really important.
In terms of the challenges, what sort of challenges? I guess there are these, as a female scientist, I guess you are facing some of the challenges as well as we also suffer from imposter syndrome, etc. So what are the main challenges that have
Yeah.
faced?
Yeah, definitely. I mean, I was thinking about this in terms of me being a female bioinformatician, and I think at times it can feel a little not isolating necessarily, because I think it's definitely changing and there are more female bioinformaticians now. But whether it's direct gender bias or not, there have been definitely times where things have happened or said, and like later on, I would question like, was that said just because I'm a woman? Like, yeah, I don't know. It's very hard to say. I'll never really know the answer to that. Nothing really terrible has happened, but it is kind of always in the back of your mind, which is just something that you deal with. But, yeah, hopefully that will just get better over time as well. And then with imposter syndrome, I've definitely had that a lot. Yeah, I think, like I said, after my PhD, when I was spread across like a lot of different projects, not really feeling like an expert in anything, I was like, yeah, what am I doing? But yeah, and then I'm probably a little bit changing fields as well. I'm like, am I qualified to do this? Like, can I do this? But yeah, I think probably most people feel like that at some stage. Yeah.
I think self-doubt is a killer, isn't it? Particularly like people just say, no, I was sort of concerned about this and doubting about this. But people look at you like, wow, that's Emma, well, she's doing fantastic research. She's doing all this analysis, all the databases. And then, it's
Yeah.
interesting how you see the two sides.
Yeah, absolutely. Yeah, I think the way that you perceive yourself sometimes is not the same as how others perceive you. But yeah, I think it's how you handle it as well. And I think just going for opportunities, even when you have those doubts, is super important. And like not holding yourself back just because you think you might not be able to do it, or you only have like 6 out of the 10 skills. I mean, you still have 6 of the skills, so just go for the thing. Yeah.
So my final question is, what advice do you give to the junior scientists? What sort of skills do you think a bioinformatician, computational biologist should have to be able to sort of progress in their careers?
Yeah, I think this is a really good question. And, I think about how different the landscape is today to how it was when I was first learning bioinformatics, especially regarding the use of AI and things like that. So, I mean, I would have said in the past, like, having a really good handle on coding and things like that is like super critical. But, now with AI being so advanced, the coding that it can do is incredible. So I would say that even though that's the case, like you still have to have some knowledge of the code to be able to debug it and understand what's going on. So I don't think we can just rely on it completely. And probably what will be super important as well is like thinking critically about the code that it's producing and how to use AI in a really effective way for bioinformatics. So yeah, I think that's something to consider. And I think even extending beyond the technical skills in terms of coding and things, I think just having that capacity for critical thinking or interdisciplinary thinking and communicating with other teams, I think those are going to be the skills that are going to be really important because, anyone can use AI to code for something, but it's like how we can apply that in the best way to get a scientific outcome. That's where we need to critical think and still talk to each other as humans. Yeah.
It is. Thank you, Emma, for that very fantastic conversation. I really, really enjoy it. And particularly because it's like you're coming across different fields and we were talking about cancer, asthma, sepsis, marine
Mm-hmm.
biology, environmental DNA, etc. So it just covers a lot of different fields. And I wish you the best in the future for you. And I'm looking forward to see what your science make an impact to not just WA, but also to the world in terms of conservation of those endangered species. I think your work is fascinating and is critically important for the human race. So thank you so much for your time and generosity.
Thank you so much for having me, Carlos. I had a lot of fun and yeah, just thanks for the invitation.
Anytime.